Hello, I'm William Reddy, and along with Emerson De Silva, we will present our findings discussing the anti-cancer activity of freshwater microalgae from extreme environments. Disclosure of affiliations, financial, and in-kind support. Affiliations. I have no relationships with for-profit organizations. Grants/research support were from Natural Science and Engineering Research Council of Canada, NSERC, and Canadian Bureau for International Education. The financial support involved here in this section/program has been received financial support from NSERC and Canadian Bureau for International Education in the form of an educational grant. Introduction. Cancer is the second leading cause of death globally. Ovarian cancer ranks sixth in prevalence and fifth in mortality among women in developed countries. Treatment options for cancer include surgery, radiation therapy, and systemic treatments such as targeted therapy, hormonal therapy, immunotherapy, and chemotherapy. Of the anti-cancer agents made available since 1944, 60% of these are derived from natural products. Studies have reported extracts from green microalgae as promising sources of anti-cancer compounds. Green microalgae synthesize and release secondary metabolites, often due to changes induced by environmental stress conditions. As a consequence, environmental stress factors such as pH, temperature, and nutrient availability can modify or enhance the production of compounds with pharmacological activity. For this purpose, we have been collecting samples from water bodies around abandoned mine sites where microalgae survive in extreme levels of pH and metal levels. Our group has initiated a screening program aimed at identifying novel pharmacological active anti-cancer compounds from green microalgae obtained from extreme environments of Northern Ontario, Canada. Methods. Sample collection and isolation. Samples were collected from Northern Ontario water bodies, including areas around abandoned mine sites where the lower levels of pH and/or higher metal levels are likely. A growth medium was added to make a 10% nutrient addition, and samples were left to grow for 2 months. Algae was then isolated morphologically and then separated by DNA sequencing. 6 microalgae strains were used in this figure. Figure 1. The strains were grown for 15 days in bold basal medium, with a pH 7 acting as a control and pH 5 acting as a lower. Then, microalgae biomasses were harvested and processed. Methanolic extraction was performed and the final extract was obtained.
For cell culture and anti-cancer activity screening, the ovarian cancer cell line H2780 was used for the screening. After 24 hours, cells were treated with 150 and 200 micrograms per ml of extracts for 24 and 48 hours. Then, the cellular viability was measured using the Hachette dye. Results. As you can see in figure 1, the effect of the extracts cultivated at low pH caused significantly morphologic alterations after 24 hours of treatment, indicating cellular death. In figure 2 and figure 3, it showed that effect of the extracts on cellular viability after 24 hours and 48 hours, respectively, of the treatment in different concentrations. The bars in gray indicate the effect of the extracts grow at neutral pH, and in black, the streams grow in low pH. Results evidenced that with the enhance of time and concentration, as well as low pH extracts, have more prominent antitumor activity. In figure 4, we compare the effect of the compounds from the same strain growth at neutral and at low pH for different times and concentrations, and we can visualize that the extracts that were cultivated in low pH have significant antitumor activity, especially with more time and high concentrations. The outcomes of that study is that the anticancer activity of methanolic extracts varied within the same microalgae genus and between the genera evaluated. The anticancer activity of the microalgae compounds was time and concentration dependent. Microalgae extracts from low pH environments showed significant anticancer activity after a short time of exposure and at low concentration. The extracts from S5, CC, LL2, and LL1 strains were cultivated at low pH, showed increased anticancer activity when compared to the ones cultivated at neutral pH, and the control strain CR showed the opposite effect. The conclusion of that study is that the green microalgae strains isolated from low pH environments in Northern Ontario showed promising anticancer activity against ovarian cancer cells. Further studies aim to identify the activity against other cancer cell lines and non-cancer cell lines.